Hi, my name is Lindsay George. I am the Director of Clinical in Vivo Gene Therapy. My laboratory studies basic mechanisms of how your blood clots, as well as a primary focus on developing next generation approaches to gene therapy for coagulation disorders. Adeno-associated viral vectors, or AAV vectors, you can think of them as a delivery vehicle to deliver a gene or nucleic acid machinery that you're interested in. The idea is you use the machinery of the original virus, but you gut the virus of its own DNA, and then you insert your DNA of interest to use it therapeutically. We encounter AAV in the environment anyways. It actually doesn't cause any known human disease, but we encounter it in the environment, and our body's response to that is to make antibodies to AAV. And so a large portion of patients that may be eligible for AAV gene therapy have what's called pre-existing antibodies, or AAV neutralizing antibodies. Largely the data would support that having these pre-existing AAV neutralizing antibodies would preclude the efficacy of delivery of the drug. We wanted to sort of tackle the root cause of what develops these antibodies, so we wanted to potentially eradicate entire B cell populations that are responsible for making these antibodies. We hypothesized actually that you had to broadly target the B cell populations, both those that are circulating in your bloodstream, but also within, you could think of them as lymph node organs, so spleen, bone marrow, etc. And actually, some of our colleagues, both here at CHOP and Penn and around the world, have a very effective way of targeting B cells, which is CAR-19 therapy, which is sort of broadly being used for leukemia, lymphoma. And we thought this might be a really effective way of getting rid of these B cells that are making these antibodies to AAV. And so we investigated that in a mouse model. The data were really quite exciting. This work was importantly done collaboratively with Bevy Adoshi and Vijay Bose, and it was very effective actually at eradicating or getting rid of these neutralizing antibodies. What I think was interesting to us is that we also similarly tried to just deplete the peripheral antibodies and B cells by counterintuitively an antibody-based approach. So you had an antibody that would draw out these B cells or kill the circulating B cells, and that was not effective. So it was really interesting because you could get rid of all those circulating B cells in the blood, but that didn't actually get to the root of the problem. So we could get rid of those where they were no longer detectable, but it wasn't effective at getting rid of the neutralizing antibodies. And I think that deserves further investigation, because it probably tells us why the human immune system is so effective at making neutralizing antibodies to AAV and why they persist, as far as we know now, documented over decades. And so the sort of future approach that I think is coming. There are some successful efforts at in vivo CAR-19 therapy, but the work continues.